In this module 1, we are going to cover macromolecules, and that includes carbohydrates, lipids, nucleic acids, and proteins. We're going to start with carbohydrates, and I am going to apologize in advance. I do not have my tablet with me here, so it's very difficult for me to write on this. I may be circling and trying to attempt to give you some additional information, so just bear with me here. The expectation is, as we go through these notes, you fill in the note sheets that you have in your notebook. If I give you any additional information that may not be on your note sheet, I would highly recommend you include that information as well. So let's get started. Carbohydrates are made up of 3 elements: carbon, hydrogen, and oxygen. I am assuming that most of you have already had chemistry, so hopefully these 3 elements are familiar to you. We are going to take a look at different types of functions of carbohydrates, but I do want to start with this term right here, a monomer. Monomer just simply means a single unit, and our monomer in carbohydrates are single units of a sugar molecule. So if we take a look at this image down here, we have several sugar molecules linked together, but when I'm talking about a monomer, I'm just talking about a single sugar unit. And we're going to take a look at some examples of single sugars. We're going to take a look at some starches, some cellulose, which are what are called polysaccharides, and so on. Most names for sugars end in OSE, and they are classified by the number of carbon atoms within that molecule. For example, if a molecule has 6 carbons, it's referred to as a hexose sugar, and an example is glucose. So if we take a look at your glucose molecule, I can count out my 6 carbons. Here's 1. Wherever there is a point here, there's a carbon, so there's 2, here's 3, here's 4, here's 5, and here's 6. So a 6 carbon sugar, an example here is glucose. 5 carbon sugars are called pentoses. An example is ribose, which is part of a nucleic acid. Again, we could count our 5 our 5 carbons. There's 1, 2, 3, 4, and 5. A 3 carbon sugar is referred to as a triose. An example is a molecule we see in photosynthesis and respiration. It's called glyceraldehyde. And again, so here's our 1, 2, 3 carbons. And you'll notice that this molecule here is represented in a linear form. These 2 are represented in a ring form. Doesn't matter, just as long as you understand where the carbons are located. We'll start with a monosaccharide. That prefix mono refers to just 1. So a simple 1 monomer sugar molecule. We're going to take a look at 3 of these. Our first one is glucose. And as we go through all 3 of these, I am going to let you know that all 3 of them have the same formula. C6, which means it has 6 carbons. If I can get this on here without causing too many problems. Sorry, that's 12 hydrogens, and 6 oxygens. And we could go through here and count the carbons. There's 1, 2, 3, 4, 5, and 6. We could go through and count the hydrogens. We won't do that here. There's 12 hydrogens, there's 6 oxygens. Glucose is found in plants. It's a product of photosynthesis. It's a simple sugar that the plant makes as an energy source. A second type of monosaccharide is fructose. Again, we could go through. It has 6 carbons, 12 hydrogens, and 6 oxygens. This monomer is the sugar that's found in fruits. A third monosaccharide is galactose. Again, 6 carbons, 12 hydrogens, 6 oxygens. This is a sugar that's found in milk products. All 3 of these these monosaccharides have 6 carbons, 12 hydrogens, and 6 oxygens. And when you have molecules that have the same formula but different shapes of the molecules, they are called isomers. I'm just going to write this here best I can. My apologies. ISOMER. Sorry, S, isomers, okay? I can go back when we get back to class, I can go back over that with you in case you can't really read it that great. All right, moving on. Let's get rid of these here. A disaccharide is when we put two monomers together, two simple sugars together, and we do that by using a process called dehydration synthesis. So down here at the bottom, I have a glucose molecule, and I want to join it with another glucose molecule. And so there's our two monosaccharides. And we're going to put them together through this synthesis process. We're going to take out an OH from this molecule here, an H from this molecule here, and we're going to take out water. And so there's that. We're dehydrating the molecule by removing a water molecule. And then in turn, here we're going to have a disaccharide called maltose, and this is a sugar that's found in alcoholic beverages. A second disaccharide, if we take a glucose and fructose, again, we're going to remove a water molecule through dehydration synthesis, and in turn, we're going to create the disaccharide sucrose, which is your table sugar. Excuse me, a third disaccharide that we can create, if I take a galactose molecule and a glucose molecule, and again, I do that dehydration synthesis where I'm removing a water molecule, we are now going to get lactose, which is a disaccharide in milk products. We're going to move on to polysaccharides. Here we have polymers of sugars, which means we have lots and lots of single sugar molecules connected together. One of the functions is energy storage, and we're going to take a look first in plants. So energy storage in plants, the polysaccharide here is starch. And so what you'll notice down here at the bottom is we have many, many glucose molecules connected together, and that creates starch. Like we find in potatoes, green beans, and carrots. Another polysaccharide in terms of energy storage is what we find in animals like ourselves. Here it's called glycogen. It's found in your liver. Now glycogen is a little bit different. We still have links of many, many glucose molecules. However, we now have more of a branching effect here. It's no longer just linear, but we have some of these molecules that are extending out from the original strand. In terms of function, now we're in structure. Plants will produce cellulose as part of their cell walls. Again, many, many, many molecules of glucose connected together. The only difference here, if you notice the direction that these molecules are sitting, they kind of alternate between one another. And then the last one in terms of structure is what's called chitin. It's found in arthropods and fungi. Chitin also uses glucose molecules, but it has a nitrogen-containing molecule that extends off of those as well. So we have these nitrogens here. That's a clue we're talking about chitin. So we're talking about the exoskeletons of insects, exoskeletons of like crabs and shrimps, lobster, and then of course the cell walls of fungi. So that's our carbohydrates. Write down any questions you may have, and I will answer those when we get back to class.